So thank you very much for the invitation. It's a real pleasure to be here. And as Laurence has indicated, I'm coming from physics, and in recent years, I moved to systems biology around 10 years ago. And recently, since I'm at IBM Research, we are working into the interface between mathematical modeling and machine learning. So my talk is going to be really a mixture. I'm going to start with some mathematical approaches, but I will start slowly moving into machine learning, and I touch a little bit of deep learning. So by all means, ask questions at any time. This is supposed to be an introductory lecture, so if you don't understand anything, just jump in and let's have some discussions. So since I won't have the chance to tell you about everything we are doing, maybe I can just give you, I wanted to. We need, it's not working. The battery is right there. The battery. Okay, so maybe I can. I think I can also point with the mouse. This is fine. Yes, so I wanted to tell you a little bit about what we are doing in my group at IBM Research in Zurich, and then I will just focus on a little bit of these projects. So we are working, our focus is the analysis of cancer, which is very generic. So we try to develop models that help personalize medicine. And we have a particular interest in analysis of single-cell data sets. Actually, we've been working quite a lot in analysis of CyTOF. Maybe some of you are familiar. It's a type of technology, single-cell proteomics, that is becoming very popular, and it's kind of very nice to see. And I will talk a little bit about it, so I don't tell you much now. So we have been working with the CyTOF data to understand different aspects of the characterization of tumor heterogeneity, the dynamics of signaling networks, etc. But we also work with traditional bulk omics, transcriptomics, proteomics, etc. And in this respect, is the second subtopic, the artificial intelligence. We have done a little bit of various different things, from text analysis to reconstruction of molecular networks to patient sample classification. So I will touch on a little bit of these projects. So more exactly, I will tell you a little bit about the dynamic reconstruction of networks, and then I will move into the artificial intelligence block, tell you a little bit of reconstruction of molecular networks, analysis of text sources. Let's see how much time we have. So this is what I have prepared, but we don't have to cover everything. So if you have questions, just let's have a discussion, which I think is more interesting. Okay, so after saying that, let me start with you a brief introduction of single-cell data and tumor heterogeneity. And I realize that maybe this is trivial for some of you, but for some of the other of you, maybe just come from a more mathematical background. So I thought, thank you very much. Perfect. So I wanted to give you an overview of why we even talk about tumor heterogeneity. So what it turns people have been realizing in the last 10, 15 years is tumors, we're talking about human cancer, are extremely heterogeneous. And heterogeneity comes in two different flavors. On one hand, you have what is called interpatient heterogeneity. That means if you take a cohort of, let's say, breast cancer patients, and you look even the same subtype of breast cancer, and you look to individual patients, you're going to realize that each one of them harbors very different mutations. But one step further, if you look at the same patient and they just took different biopsies from the same patient, you will also discover that there is a lot of heterogeneity. So why this? Okay, here. So the second one, which is the one where I'm more interested in looking at, so where it comes from the fact that biopsies is a collection of cells with very heterogeneous backgrounds. So here you have a collection, usually when you take a biopsy, this is a finite volume, like thousands of cells. So you will have a sample of healthy cells. You will have a mixture of epithelial, vascular, stroma cells, immune cells, etc. Each one of them take into account is a stochastic machine. So there are normal probabilistic process that just result in some randomness. So you have a lot of heterogeneity just from the normal healthy processes. But on top of it, you have all the cancer processes. So you have a lot of different cellular cancer cells. Each one, they are going through a process of clonal evolution in which they are constantly replicating, acquiring new mutations, and then increasing the heterogeneity. So all of them together means that you have cancer samples are extremely heterogeneous. So what is the problem of this heterogeneity? Of course, if you have in the first scenario, so suppose you have an extremely homogeneous population, which each cell have exactly the same expression level of different markers, then of course you don't care about single cell data. You just take one sample, you compute the average, and then you're done. In real sample, you have more like this one. Maybe the red marker here is a biomarker that might be important to distinguish certain population, but then you take the average, maybe you are just missing difference of populations. So because of these challenges, in the last, let's say, 10 years, there has been, maybe a bit longer, there have been an explosion of single-cell methods. And right now we have a bunch of transcriptomic single-cell methods, single-cell DNA sequencing, single-cell proteomics. Now we are moving into special single-cell methods. So there is a plethora of methods out there. The nice thing of this method is because now you have information at the single-cell level, you can look. First of all, you can look at subpopulations. You can just simply characterize, okay, give me clusters and tell me whether this cell is highly expressed is this marker or this other marker. This is just the first question that is interesting. But then also some of these markers might be biomarkers, because if you are able to decide that this population has a higher metastatic potential, then you might want to target this particular population. But then you can start looking at the second level of analysis. You can try to unravel what is specific of each subpopulation. So maybe this subpopulation has a particular mutation that makes them more aggressive because it's disrupting this pathway. So all this is only possible once you have access to single-cell data. So as I mentioned at the beginning, we've been working with this type of data, COSIFER, which might be familiar to some of you. COSIFER is a short name for mass cytometry. So basically, it's a combination. It combines flow cytometry, which is a technique that has been around for quite a number of years, in which you just basically isolate cells, and then you select 4 or 5 markers, usually proteins, and then you label them using an antibody with some fluorophore. So it's a particle that you can excite with a laser. And then you just isolate one by one the cells, and then with a laser you are measuring the expression. So it works quite well. But because you are looking at, you are using fluorophores, these are particles that have a quite wide spectral band. So that means you cannot look at many of them simultaneously, because then you start seeing a spectral overlap. So because of this challenge, people have sought to combine flow cytometry with mass spectrometry. So how does it work? So the idea is the same. You isolate cells, you select a number of markers, and then you label them through antibodies. The difference is now, instead of using traditional fluorophores, now you use heavy metals, which can be detected instead of with a laser. Now you use a mass spectrometer. And the advantage that you have now a very narrow spectral band. So now the advantage is, instead of looking at 5 markers, as people were doing before, now you can look at 40 or 50. And well, the theoretical limit is much higher, but the reality is today people are measuring around 40, 50 markers simultaneously. And it might not seem a lot, especially if you look at single-cell transcriptomics, that they measure thousands of genes, but think about it, like it's mass spectrometry, so it's cheap, and you can profile thousands of cells, even millions of cells in a relatively small amount of time. So this is a power that still single-cell transcriptomics does not have. So okay, and now I have introduced the data we've been working with. I wanted to tell you a little bit of how you can use this data to reconstruct the signaling dynamics of networks. And actually, this project is very nice. That is the first project I'm going to discuss about, because it really links well with what Herbert has introduced this morning. We will see now the similarities in a second. Let me first tell you a little bit of introduction. So the problem of network reverse engineering of networks, as probably you have heard before, is that this usually is a difficult ill-posed problem, in the sense that in a traditional setting you will have a few measures. I don't know how many you have measurements, maybe you have 10, 20, depends on how expensive is your system to characterize. And then, but usually you have the challenge, you have many more variables than experimental points. Just if you think about the standard basic problem, suppose that you would like to reconstruct the network of interactions in a transcriptomic experiment. You have 20,000 genes, and maybe you have 10, 20, 100, 1,000 experimental points. Still, this is a very ill-defined problem. Of course, people do simplifications in the sense that you don't consider all markers, you consider a subset. Then you also do some heuristic approximation about the kinetics. And then you do certain approximations, you kind of neglect stochastic effects. And then in the end, you can somehow define probabilistic approaches to reconstruct these networks. But the important thing to remember is that in the end, you're never going to get an exact network. You're going to get the most likely network for your data. So just keep in mind here, I'm coming from a probabilistic perspective. So what you get at the end of your inference problem is not the exact network. It's the most likely network. So yeah, exactly. We cannot infer an unique solution, but we can infer the most likely solution. So actually, Herbert this morning was really giving you a very nice introduction about the different probabilistic approaches. And the second approach he mentioned, he did not mention this word, but he was talking about the graphical lasso. So if you remember, he was talking about the inverse covariant matrix. So I will talk a bit more about it. So, but first of all, I wanted to present the challenge of network, one of the challenges of network inference. So suppose that you have, this is a very naive problem, so you have this toy model, so this will be a system with 4 genes, and you have some experimental so time series data, and you would like to infer the connectivity. Here I'm not even considering causal network, these are undirected edges, so I'm even simplifying further the problem. So suppose here in this experiment you have 3 time points, time T1, T2, T3, and on each one of them you are measuring the correlation between markers X and Y. So you can see, like in this model, we assume that Y and X are related through this logistic function, which is a quite standard assumption. Basically, if you come to the biological meaning, what it's telling you, there is a regime where basically there's not enough amount of X to activate the expression of Y. Then when you pass this activation threshold, you have somehow a linear response, but then you reach a saturation regime. So this is a function that's a functional relationship that is usually standardly assumed. So the problem with this approach, I suppose that you are unlucky enough that you measure the time 1 is here, time 2 is here, and time 3 is here. These are the 3 times that you are measuring. So here you will conclude, because here the curve is rather flat, you will conclude that there is no correlation. Then you look at here, you think there is a linear relation, so you will infer there is some correlation. And then the last time, you assume again that this is a kind of flat curve, so there's no correlation. So when you infer your network, actually this is your inference. You think that here X and Y are not correlated, here are correlated, and here they are not correlated. And actually, you think about it, this is wrong. X and Y are functionally related, it's just there are certain regimes in which the activation, the functionality is not active, but they are always related. You can think about it as you suppose that you have imagine that this network is somehow like a road map of a country. So sometimes of the day there's higher traffic, so a larger number of cars. So you are just trying to infer the road map by looking at the traffic, the number of cars. When you look at midday, you will see high traffic, so you infer there is a road here. But it's not that the road is removed at night, it's just there is less traffic, but the road is still there. So this is the same approach here. The connectivity is always there, it just sometimes it's not active. So how did we look at this problem? So this is when I'm connected with what Hervé explained this morning. So this is a very brief introduction to graphical models, and because he already talked about it, I'm not going to spend a lot of time about it. Just telling you that these are again very simplistic graphical models. Here I'm just talking about 3 nodes, but yes, you get the idea. You can make it larger. Graphical models, they can be directed or undirected. And, and when you talk about graphical models, the concept of conditional independence that Herbert discussed quite extensively is one of the key concepts to infer the topology. And actually, the approach we're going to be using here is what is called the graphical lasso. So basically, if you are willing to assume that the data is Gaussian, so this is the main assumption of this approach, your data has to at least be Gaussian enough. So if you can assume that your data is Gaussian, there is a nice theoretical result that tells you that you can just simply compute the covariance matrix and then take the inverse. So this is also called the precision matrix. And this matrix just gives you the topology of your network. Here I'm talking only about undirected network. So this result only applies to indirect networks. So basically, you compute this precision matrix, then you look at off-diagonal elements, those that have a 0 or close to 0. That means that the links are conditional independence given all the other networks, all the other nodes in your network. So this is the basic assumption of a model for reverse engineering that has been quite popular and has been used a lot in the literature, what is called the graphical lasso. So graphical lasso basically takes this result that you can infer the topology of your network given the inverse covariance matrix, and then you just use a probabilistic approach in which this term is only the maximal likelihood of the inverse covariance matrix. And then you add a penalty because you know that your network should be sparse. That means if you don't add this term, what you will get is, as you asked this morning, what you will get is this network which is densely connected. You will always get a non-zero coefficient for each edge in your network. But you know that biological networks should be sparse. You know that they typically follow, in many cases, a power law. So because you know that, you can infer here, you can impose here a sparsity such as you are forcing many of the coefficients to shrink to zero. And that will give you, of course, you have to fine-tune the level of sparsity, but assuming that you compute this sparsity at the right amount, you will get a network with only a few coefficients are not zero. So you will get this network that hopefully it will reproduce your data. So this was published in 2008 and has been heavily, quite a lot, very used in the literature. Many people have tried to reconstruct graphical models using the graphical lasso. What are the problems? Well, the first one is as I mentioned, well, not problem, limitations. The first ones, as I mentioned, you are assuming that your data is Gaussian, which may or may not be true, and well, sparsity. But the main challenge in our case is it does not take account the temporal component. And as I explained before, given these logistic functional interactions between different genes in the cell, this may be a challenge. So let me show you some real example to just convince you that this is really a problem we encounter when we working with the real data. So it's not just a theoretical concern. So these are some experimental curves that we computed using site of data. So here we are looking at these two markers. P stands for phosphorylated. So this is ER key, and this is 90 RSK. So these are two markers that are involved, they play an important role in breast cancer. And you can see what I explained before. Here you can look at the conditional density. So in these two curves, actually, when you compute the conditional density, you get, well, you can see a little bit of a slope, but it's mostly flat. When you compute a threshold of pi value, then you will get that here these two markers are not correlated, except for the last point. So as you can see here, even though, according to the literature, we know that these two markers are correlated, the graphical lasso, when applied independently to each time point, is going to tell you that these two markers are only correlated at the last time point. Okay, so what did we do to address this challenge? So actually, the idea we had is kind of simple, but it works remarkably well. So coming back to the likelihood definition of the graphical lasso, remember that was the maximum likelihood estimator. That was the sparsity constraint. So we just thought about putting what we call a smoothness constraint. So basically now we have a second parameter. This is a price we pay. But what we are saying is we know that the topology of the network does not change substantially, should not change too much across time points because the system is the same. Maybe you are stimulating some pathways. You are kind of the traffic of certain roads is increasing, but the topology itself should be rather stable. So here we say this is basically data measured in time point I, and here is the previous time point, and we put this sparsity, this constraint saying that we don't want big changes. We are penalizing big changes. And that's about it. The rest is the standard graphical lasso. We only use this smoothness constraint. Okay, so how does it work? So let me show you, of course, with a new method, the first thing you have to do is to benchmark it in some synthetic data where you have a good control of what you are putting in the system. So this is what we did here. And sorry, I don't have labels, but the red is our approach that we call dynamical graphical lasso, and blue is graphical lasso. And as you can see, well, several things. Well, first of all, this is a synthetic data set where we assume there were 30 nodes to replicate the site of experiment. And then we simulated 20 different time series data sets. So when you see the standard deviation, these are around 20 different data sets. So the first thing you said that, well, in the first time point, still dynamical graphical lasso performs a bit better, although not substantially better, but this is understandable because it's the first time point, so you don't have information on a previous time point. But for subsequent time points, you see that we are doing substantially better. And not only that we are, as well, this is the area and the ROC curve, where you are looking at the rate of for positive, true positive versus the false positives. And not only we are doing the curve is higher, so that means we are doing better, but also if you look at the standard deviation, actually they are substantially smaller. So actually, at least in synthetic data, dynamical graphical lasso outperforms graphical lasso. So coming back to the example I showed before, this is the same example, the same two markers I was speaking about before. And you see that now dynamical graphical lasso gives you this connectivity at all time points. And of course, you get the connectivity with different confidence levels, because here, obviously, you have less confidence to make your prediction. But still, you are inferring across a stable edge across all time points, which is a very nice feature. So to show you, I wanted just to, before wrapping up, I wanted to show you the result of like a full network inferred with this method. So this is the result of a site of experiment. I think we had here 30 or 36 markers, I cannot remember. So the edges with the thick solid line, they are edges that are stable across all time points. You see that, let's say, half of them, they are stable across all time points. And the ones with dashed lines, they are ones that are changing. But even the changes, as you can see here in this experiment, so in the experiment, each row will represent an experiment at one particular time point. So this is time 0, then 5 minutes, 10 minutes, 15 minutes. And here, sorry, we are perturbing the IFN gamma signaling pathway. That's a signaling pathway that is important for the signaling of breast cancer cells. So you can see that even though there are changes across time points, overall the signature is quite stable across different time points. And then here, what we are showing here to validate our experiment, our predictions, what we did is just inhibit certain of the nodes to validate certain of the predictions. So here in this network, here I'm showing the inhibition of PI3K kinase. And actually, when we inhibited this protein, we actually saw that some of the edges that we predicted to be present with the dynamical graphical lasso were lost. So that was a nice validation. So this was published this year in scientific reports. If you want to look into it, please feel free, and if you have questions, come back to me. Okay, so this is, I wanted to move now to a more, starting to move more and more into the machine learning, deep learning. So as I said at the beginning, please interrupt at any time if you have questions. Just, there are questions for the time being? Okay, not.
We assume that all these lines disappear due to the PI3K inhibition at once. Is it so unspecific?
So you say why we only have, because here we only have one time point. So here we cannot have, for the inhibition, we only did one. Oh, no, sorry.
The network in B is much smaller than in A.
You're talking about the number of predictions. Yes, so I imagine, yes, that when we inhibited, some of the connections were lost because we are inhibiting this key component. This is a receptor. This is at the beginning of the cascade. So many of the interactions are meant to disappear because of the inhibition of the experiment.
You also mentioned interactions that are not directly connected to this PI3K.
Yes, usually biological experiments, they are never as clean as you only lose the signature you are trying to look at. So of course, there are always some perturbations that spread. And mass spectrometry, although the nice feature is you are measuring thousands of cells, so you can always, you have a statistical robustness, you also have a high noise. So there is, it's not that we only lose interactions associated with PI3K, we also lose other interactions. But I think overall the signature was quite clear that the main interactions that were lost with highest confidence were the ones related to PI3K kinase.
And you can't maybe even try to find if it can be forming complexes with others, and maybe that's why you are losing other interactions that were not directly related?
Sorry, your question?
Can you identify complexes by these losing interactions that are not directly linked to PI3K? So can PI3K be in a complex with others? And is these other ones the ones that lose the interactions that are not
So all these are signaling proteins. That means they are binding to each other, and we are measuring the phosphorylated protein. So usually the one that is in an active form. So, but this is with the site of experiment. Actually, you are only this interaction. These are phosphorylation events. They are rather fast. They are very fast interactions. So usually, you have an equilibrium in which you are binding and unbinding. So I don't think this is not that the protein is just bound and sequestered for a long period of time. So this is because it's very, very fast binding and unbinding kinetics. You can assume that you have an equilibrium where you can detect in the averages. Any other question? Yeah?
Can you comment on the Gaussian distribution assumption for the cycle? And how difficult is it to choose the lasso and the smoothness parameters?
So about the Gaussian distribution, this is one of the essential facts of this theoretical proof is based on Gaussianity. Sometimes when you do, because for instance, site of data is everything but Gaussian. Actually, well, it doesn't matter. It's a distribution that is quite skewed with very long tails, because sometimes you have markers that are very highly expressed. So what we did, and actually it works for this project, you can do some transformation, and here people traditionally work with hyperbolic arcotangent. I don't ask me why, but something that works, and usually brings, still is not Gaussian, but it's more or less symmetric and resemble a Gaussian. So actually, you don't need to have exactly Gaussian data, but something that resembles a symmetric distribution. And if you have this, it works. It works in this situation. If you have something that is completely asymmetric, we have not tested it, but probably you can have challenges. And your second question was?
The tuning of the parameters.
Yes, so for the tuning of the parameters, this part I'm not discussing, but actually we did, the students, well, not me, the student who was working on it did a very nice statistical approach to modify the VIC criterion, the Bayesian information criteria for this approach. So, I mean, we can discuss later on. I mean, all the details are in the paper, but basically they did an extension to that use for these two parameters using a very statistically very robust approach. So not going to the details, but I'm happy to discuss afterwards. Okay, so then I'm moving to the next project. So as I'm saying, I'm moving now more into the machine learning realm of life. And here, when we started this project, it was a few years ago, we were working on an European project that was focused on prostate cancer, and we wanted to develop personalized medicine approaches for prostate cancer. So when we started working on it, actually the first project, the first problem we wanted to solve is just how to automatically reconstruct data. So here, in the previous project, we were talking about a very concrete, well-defined data set, and we want to infer signaling networks. Here we come in completely different realm of research. Here we have a lot of big data sources, more or less noisy, with different types of distribution, types of noise, and we want to do is a reconstruction network that can be automatically applied. And that was one of the main concerns for us. We wanted something that can be done automatically with very or none at all human input. So I wanted to present two different projects here. The first one is, we call it Interact. It was published last year. And this is for analysis of text. So let me tell you a little bit, and I know this might be a bit too far away from most of you. You come from the mathematical domain of life, but I think it's nice at least you get an overview of what people can do in the more AI side of the dark force. Okay, so when we started working on it, what we wanted, again, we were focusing on prostate cancer, and we wanted to develop a tool that could extract, the goal of this project was to predict a network of protein-protein interactions from the literature. So this is, now we are getting into the domain of natural language processing, and there is a bunch of, many people working on it for a number of years, so there is a bunch of methods that have been developed. So of course, we did not want just to add another method to the big pile of methods. But we came with slightly different constraints. So many of the methods that have been developed to extract a relationship from text, they have a few requirements. And the first one is they usually are rules-based methods in the sense that somebody has to spend some time annotating text, so you have to read the articles. And if you are trying, for example, suppose you try to predict the phosphorylation interactions. So you have to train your algorithm. You need to give a lot of annotated text to this algorithm where you define this is a gene, this is a protein, this is a relationship. So this is usually human annotation that takes many hours. And this is a heavy requirement of these methods. Also, very often you need a language model that is some kind of, you can think of as a probabilistic distribution that tells you how likely is to have the next word given that you have this word in a sentence. This also is highly context specific and requires a lot of time and effort to develop. And what we wanted to do is something that was as unsupervised and possible, in that way that you can apply today to prostate cancer and tomorrow to finance if you want. So something really generic. So what did we come with? So at the time when we started working on that, people were paying a lot of attention to a technique that is called word embeddings. And I'm going to tell you a few, but I have 2 slides about it. So about word embeddings. Word embeddings is simply a very fancy term to just a technique, any type of technique to transform a word into a vector. So basically you have your word and you have a high dimensional space. Typically people work with a few hundred dimensions. We work with 500. In that such way that you have your text corpus and then each word is going to be a vector. And when we started working, it was maybe 3 years ago, actually, this project, this word2vec, had been recently published. And what this author did was very interesting. So they developed this method. In the next slide, I will give you an introduction of the method. And basically what they did, they took all the articles from Wikipedia and they applied this word embedding. And actually what we did, so they have at the end, after this application, you have your vector space with 500 dimensions, and each word is a vector. So they tried to, one of the first things to try, the test we did, was just simply they took this collection, I don't know if you can read, but it's a collection of countries, and here a collection of capitals. And they just projected them into 2D. And this is, the important thing, this is unsupervised. So you have never told your algorithm what is a country or what is a capital. The algorithm has understood that by itself. And the nice thing is you can see here this is kind of horizontal alignment. So when you project, you have that China more or less aligns with Beijing, Russia with Moscow, Turkey with Ankara, etc. So again, this is completely unsupervised. So just the algorithm has understood that there is a similar relationship between these 2 words and have placed them into positions in the word embedding space in such a way that this relationship is preserved. And the same can be seen when you look at other examples, other type of relationships. So here, for instance, not sure if you can read, but here is king and queen, man and woman. So when you look at this gender transformation, they also seem to follow a similar transformation, algebraic transformation. And the same for maybe verbal tenses, like from walking you go to walked in this direction, from swimming you go to swam. So actually what the people started to realize that actually the word embedding is placing words in a position that makes sense to preserve the semantic context of this word. So that means that given this position, you can try to make algebraic operations on the word. For instance, you ask, once you have trained your word embeddings, you can try to ask what will be the word. So you take the word of king, you subtract the word man, and you add woman, what do you get? And then you can say that you can get a win. So this I know is, but people started seeing this, they got very excited, and we tried to apply it to prostate cancer publications. But before I go into what we did, I wanted to tell you a little bit how it works. So basically, this is an approach that uses deep learning. It's not a very deep network, it's a very shallow 2-layer network. And what it does is just you just go through your corpus of the text, and then you define a target word, and for each target word, you define a window context. This is something you define the size of this window. In this case, you have 3 words to the right and 3 words to the left. And then this is the input you are giving to the network, and then you're asking the network to predict, given a word, in this case P10, to predict the most likely neighboring word. And you train this well, so you have enough publications. Actually, by doing that, the network is automatically learning this vector representation of each word. Okay, so this is what we did. So far, we use this word2vec application. So far, we have not done anything new. But what was the challenge we found? So this is the idea. So here, you take the corpus of publications, and then you get this vector space. Yeah.
Just have a question. When you say 500-dimensional space, you have some interpretation of the dimensions, or because then you said you can subtract and add. So is there some meaning to these dimensions?
No, so the dimensions don't have any meaning, and the subtraction and addition is defined in any vector space. So this is like the dimension is something an ad hoc parameter that you define. We use 500 because it's what people typically use. But then once you have defined this number, that should be high enough, then you can do any algebraic operation that is defined in a vector space.
Yeah, so it's not a number of concepts or something.
No, I don't think it conveys any particular meaning. Maybe you could try to see if it aligns to certain concepts, but Probably the meaning is a bit more convoluted than that. Okay, so as I was saying, until here we just use word-to-vec, so nothing new. So then the idea is like you trust this word embedding philosophy. That means that if you try to infer a relationship between two proteins, you just have to look at this word embedding space and try to look at the distance between two proteins. If two proteins tend to appear together in papers, they will be positioned in locations that are close enough in the word embedding space. So you could just look at distances. What is the challenge of that? So actually it has been demonstrated that, the concept of distance in high-dimensional space, for instance 500 dimensions, is a bit trickier than in 3 dimensions. For instance, if you use Euclidean distance does not generalize to high-dimensional space. And there are a very nice paper that show that you can get very counterintuitive results. So for instance, you assume that Euclidean distance, sometimes words that should be close to each other appear very far away. So it's not, it has a very non-trivial behavior in high-dimensional spaces. So Euclidean distance is ruled out to measure the distances. People have also been using cosine distance, in which you have a vector, just compute the cosines, and you assume if they are close to each other, you get a small cosine. It works a bit better, but it also has a bunch of problems. And also the challenge is that you really are not taking advantage of the power of word embeddings, which is that it just really captures the semantic similarity of words. So we design a completely new approach, that's what we call interact. And actually what we do is, it's not very complicated. It's what this cartoon is trying to tell you. So basically we started with the word embedding. And first we cluster words in this word embedding. So here each cluster is what I call a different color. And then you look in this space, you look at 2 particular proteins, for example, AKT1 and MAP kinase 1. So instead of just looking simply at the distance between these 2 guys, you can try to look at all the neighbors in a given radius that you have to define. So for example, in this particular case, for AKT, you can see how many yellow neighbors, how many red neighbors, how many green neighbors, etc. And this is going to give you fingerprints of what are the more likely clustering associations for the neighbors of this word. And you can do the same with MAP kinase, and now you can compute distances between these 2 distributions. You can see this as a distribution of neighbors. And what we found is this is much more informative than rather just simply looking at the distance between these 2 proteins. And if you think about it, it makes sense, because actually you are not only looking at the position, but you're also looking at all the neighbors of a particular vector, which conveys much more information. And finally, based on the comparison of these 2 fingerprints, you predict or no an interaction. Okay, so let me show you some results. Actually, what you can see here, this as an example, what we did is look at the pathway for prostate cancer. Of course, we are interested in prostate cancer. So we took the definition according to the Keck pathway. This contains 87 proteins. 65 were in our word embedding, because sometimes we lose a few proteins because if they don't appear with a high sufficient frequency, they are discarded. So we don't have all the proteins. So we had 65, and then this is the plot that we got with interact. And actually here again, I'm showing you again this rock curve where I'm comparing the different methods. So the red curve is interact, which you can see clearly outperforms the other methods. So here in these 2 curves, you have both the correlation and the cosine distance. They are very similar because, of course, there is a simple relationship between correlation and cosine. And here, this is the Euclidean distance. As I said, it performs close to random. As I said before, Euclidean distance has a very anomalous behavior in high dimensions. So this was a very nice demonstration that actually you can do a lot of work. Starting from the word embeddings, you can do a lot of better inference just by changing the way you infer relationships. So any questions so far? Yeah.
Yes, sorry, maybe I missed that. What's the gold standard you're looking at to calculate?
Sorry, yes. We compare with string, which has a number of problems because we selected human interactions, but still, string is a collection of different experiments, different tissues. We only selected high confidence interaction in string, which I think you have a threshold of 0.7. Still, it's not perfect, because we are mixing very often different tissues and diseases, but the problem with always validating network inference approaches is that you never know the gold standard. So this is a problem everybody's facing.
Yes, moreover, I wouldn't be surprised if your true positive rate doesn't go too high, because you're looking at a subset of texts. So, yeah. You're not covering everything in one stream, so in the past.
Yeah.
Maybe you do.
Yeah, no. Ideally, it would be nice if we could compare, we have a gold standard just for prostate cancer, but as far as I'm aware, there's no such a thing. So here we have to compare with the average. That's sure. I think the true positive probably is higher, but it's at least we are comparing the same for other methods. Yeah?
And you tell us also in your previous slide, how did you define the number of clusters that you have?
The number of?
The number of clusters that you have.
Yes, this is again another hyperparameter you have to tune. I think here we use 2000. So you have to tune it. There is not a rule you have to use. It has to be high enough to cover different. So each cluster will correspond to a concept of words. So it has to be high enough to cover the diversity, but not too high that then you start running into problems that you have clusters with very few data points. So here we use 2000. I think there was trial and error. There was no. But actually we did not, now I remember. So we did quite an extensive analysis of the effect of different parameters, hyperparameters, later on for this paper. And actually we found that the predictions were actually quite robust across a large range of hyperparameters, which was quite a nice feature to find. And also interestingly, so one of the reviewers complained that we are only looking at abstracts instead of the full text. So just as a demonstration, we run the same pipeline using the full text of the articles, those that are publicly available. And actually, we found a slightly worse performance using the full text, and apart from just having much larger running times. But actually, when we thought about it, it actually makes sense, because the abstract usually presents a condensed, summarized version of the main results, where in the main text you can have sometimes digressions. People are talking about things that are not really related. So actually, the main message is that things are quite stable across parameters within the reasonable ranges of parameters.
How long do you need to run these algorithms more or less?
So if you want to run the word embedding or just interact, so the word embedding, none of them is super, if you run it only on abstract, it's a question of hours. But once you have the word embedding running interact, it's not very heavy. So actually, I don't have the screenshot here, but if you're interested in running it, we have, for all the tools we develop, we have web services that are freely available, so you can use it. And we have already some pre-computed embeddings. So we computed embeddings for different cancers, I think 10 cancers, breast cancer, lung, the most common, for which we found a sufficient number of publications. So the embeddings are already pre-computed, so you can just play around and build your network with your proteins of interest. So this runs in question of seconds, or maybe a minute or two. So just you can go to our web page of just look for IBM Research Zurich, and then you will find us, Computational Biology.
You built a network for different types of cancers. What did you differently, and what did you make out of the difference for different cancers?
For the network itself.
If the network that you have in the next slide is different for different cancers, what does that mean?
Yes, well, the network is different because, So for the network is different because for each cancer where we are plotting, here we just selected the 65 proteins that are important for prostate cancer. So for breast cancer, you will have a different subset of proteins that are important for breast cancer. We did not try to do, this would be a nice idea, but we did not do it, to try to build a common network for all cancers and look at the performance. We were more interested in finding the pathways specific for each cancer. What we've...
In a way, so it's a representation of the signaling and gene expression networks that are underlying it, which, as you laid out in the beginning, must be somehow stable in the cell. Or
yes, it's stable. So the topology, you're right. So the topology, I don't expect it to have drastic changes in the topology, especially in cancer. Most often you have rather a loss of function rather than a gain of function. So what I will expect you will get is at most certain links are missing, because maybe since certain mutations have been activated certain proteins. So I would not expect high differences. It's just the nodes that we were using here in this comparison are different, but the network should be rather similar in this context. What we found is the confidence of the predictions, not surprisingly, depend on the number of papers, and that changes drastically, even at one order of magnitude versus prostate cancer or other more rare head and neck. Maybe there are not so many publications, so then you have lower confidence in your predictions. Okay, so if there are no more questions, I'm gonna leave text on the side, and I'm going to come back to this plot. So as I showed you at the beginning, we wanted to build this automatically this network where we have 3 pillars of information. The first one was publications, and now I want to move to this pillar, molecular data sets. And then I wanted to tell you here about this other project that we are in the process of publish right now. So here, again, our goal is always the same, to build automatically this network that they give you interactions between different components in the cell, in our case, proteins or RNAs. Here the difference, instead of coming from publications, here we're coming from molecular data sets. For instance, here we have used a lot of TCGA, probably you know. So this problem, again, is not a new problem. People have been working on network inference for quite some time. And actually, so here again, we did not want to develop just another method for the sake of developing a new method. So when we started working on it, we looked into many of you know the DREAM challenges. That are these computational challenges that happen, I think, twice a year, where there is a question asked to the community, and there is some data sets, and people have to develop the best algorithm, and there is a winner. That's the competition. So these challenges are gathering a lot of attention, and many people participate in them. And there was one challenge a few years ago that was about reverse engineering. So try to decide what was the best network, the best algorithm to predict a network of molecular interactions. So it was DREAM Challenge 5. So the challenge was run, there was a winner, but actually what was more interesting rather than the winner was what they call the wisdom of the crowd. So basically what they found is that even if you pick the best method, if you combine all the methods together and you build what is called the consensus network, so that means it's this network where you have kind of aggregated the predictions of each possible network, this consensus network performs at the same level as the winner method. So this is the wisdom of the crowds. The crowds know better, or at least as much as the best performer method. And this is you think it's rather impressive in the sense that it's a really important result, because I tell you, sometimes you might not know what is the best method for your data. And this is an important point, because there is not a single method that performs best across all data sets. Usually the choice of method depends on the data set, the type of noise, the number of samples, the number of variables, etc. So there's not a winner method that always wins. And very often you might not know a priori what is the best method for your particular problem. But what this result is telling you is that actually you don't need to know what is the best method. You just can take a consensus of different approaches, and you are, well, this is not a theoretical result, but let's say you are almost sure that you are going to converge to something, if not best, at least very similar to the best performing method. So actually, following this approach, what we wanted to do is just to build what we call COSIFER. It's a service for this consensus network inference. So what we did is just look at this challenge, and we took one of the best methods, because we could not implement all the methods, because that would be a massive effort, and many of them don't have even scripts that we can easily take and implement. So we just took a collection of the methods that were best well known in each category. So we took some methods based on correlation, like Pearson and Spearman. This is rather simple. Some of them based on mutual information, like arachnes, you might have heard of. Some methods based on regression, like Tigres or graphical lasso that I introduced before, and then other methods that are harder to categorize. So we took 10 different methods, and for each one of them, we kind of implemented into this pipeline where you can, now this is, again, there's a web service, you can upload your data, you can choose the method, or all of them, and then you can get your consensus result. So I wanted to get a little bit into the results, but before that, I wanted to tell you a little bit about the methods. So, I mean, some of them are probably you're familiar with, like a correlation, I'm sure you know it. Mutual information, arachne, I'm sure you know it. But there are other methods, like for instance, GNA3, that was the winner of, maybe it was the winner of this challenge, of another associated challenge. So this is a rather interesting method. What they do is just, you have your data set, and you want to predict your network of interactions. So they use an ensemble of trees, but instead of just doing it once and getting a prediction and that's it, you are done. What they do is just an ensemble method. So you take a subset of your data and a subset of your genes, and for each one of them, you find an ensemble of trees. And then they have a gene ranking scheme, and then prioritize, they give a confidence to each edge, and then they get to a final ensemble prediction. So it's another idea, you think about this, again, the same concept of aggregating different predictors to get to a higher confident result. Another method that is also quite interesting is, for example, Tigres. This is again is a linear regression, in which you do like a typical minimization with a penalty, a lasso type penalty, in which you kind of try to shrink most coefficients to 0. The only difference in Tigres is they add also this stability selection criterion, which is kind of a nice feature, where basically what they do is again a little bit of ensemble method. So they basically rerun many times this lasso regression, but they give a different weight to each particular gene in order to just get different versions of the lasso, that the regression that they then aggregate. So it's a little bit trying to just go through this, to have these ensemble methods that are always shown to perform better. Anyhow, so how this, to the benchmark how we were performing again, because this is against a network inference and we don't know the ground truth, so we generated some synthetic data set. So in this case, we use GNetWeaver, which has also been used in many of these challenges. So it's a very nice approach to generate synthetic data set. The idea is that you start with a real network. So in that case, they use something from Cerevisia. And what they did, so the idea is like you don't want to start from a random network that might have the wrong topology or might not have the right the topological features that resemble your network. So you start from a real network, then you extract submodules. They have certain rules to transform these submodules into differential ODEs. If they use exactly, they have certain rules in which when you have a particular gates, as in maybe two nodes getting into a gene, they can just decide that this is an additive or a OR gate. They have certain ways of doing it. And in the end, you get an ODE system for each ODE, from each submodules. And you can use that to generate time series data. And from that, you can just run your network inference and then compare to the ground truth. So let me show you some results. So here, first of all, about the synthetic data. So because when you're generating the synthetic data, you can play with different sizes of the node, the network, different numbers of variables, different numbers of samples. And also you have in GNetWeaver, you also have the possibility of playing with different types of noise. So in the end, for each network, we had 48 different settings. That's why you have here these different distributions of prediction scores. So here, what we're having here in the y-axis, we have the area under the curve, under the rock curve that I showed before. And this is the prediction of each method. And there are some interesting results. As I showed before, so for instance, here you have the regression methods, graphical lasso and Tigres. They perform well. Graphical lasso does not do very well. And my guess is that graphical lasso is a method that really is good for sparse data sets, and our networks were not particularly sparse. So that's why the reason they might not perform so well. This GRF also does not do very well. And then, but here, for instance, the ones that perform systematically good are the ones based on mutual information. But then what I wanted to show you is the ones in green. So these are the ones that we call the consensus network. And as you can see, they are all of them at the same level as the top performing methods. So here we designed 3 different consensus strategies. So we have what we call, well, here we call it WOC, is the wisdom of the crowds, it's the acronym. So the simple, naive approach is that you take the average of the 48 simulations, that's it. Then you can also instead just take the average of only the non-zero predictions, because in many cases you also have 0 predictions, so then you discard those. And this is a more sophisticated averaging, SUMA, that has been recently published. So SUMA, it does a weighted average in which you take into account the confidence of each prediction before making the average. So there are different ways in which you can build the consensus network. But the important thing is in all 3 cases actually, well, maybe work a bit smaller, but you are at the same level as the winning method. So this, as I showed at the beginning, this is the wisdom of the crowds. You don't need to know what is the best method. You can just use different methods as different as possible, so you get predictions. You have a combination of complementary methods, and then you integrate them, and you are almost guaranteed to perform at the same level as the top performing method.
Maybe I missed the point. So in the consensus method, are the best method included in it?
In this simulation, all methods are included. So even those that are not performing that well. And this is the nice thing, even you are including things that might not be very robust, in the end, if a method is not performing well, it might predict at an edge that is not correct, but it will be the only method predicting this edge. So when you take the average, actually it's going to get a low weight.
You could also, once you've done that, you can also take the best method.
if you know what is the best method, but usually you don't know the ground truth, so you cannot judge.
No, of course, but once you benchmark the methods enough, it would be nice, because obviously the idea of combining different methods is appealing, so it would be nice to have even better result by combining these.
Actually, yes, if you could know what are the best methods, yes, you just pick up the best and you are done. It's just that in general, even for a new data set, I would not trust that for a new data set I have enough intuition. Even if I have worked with RNA-seq data for 1,000 times, I would not trust that this experiment is not different. There is a lot of technical effects and batch effects and many things always influencing the results. So I think what this is telling you is that you don't need to choose. You just take everything and you perform at the same level at the best methods. Yeah?
Depending on the nature of the input data, you probably expect some methods that are better than others, especially for correlation methods, for example, expect some sort of continuity in the data, so in case of very sparse data, probably correlation methods will perform very poorly. If you have very complex and non-linear relationship between the cosmology random forest, you will probably get to better relationship. So the point is, did you try, for example, with very sparse data set, ensemble method, random forest method, as compared to method including especially correlation method? Did you see any differences?
So sorry, your question is if we tried?
So in the case of very sparse data has been used, so if you consider all the ensemble methods with and without correlation methods, for example, as compared to random forest alone, do you still have an improvement by having a correlation method?
So we did not try specific methods for specific data sets. I mean, you are right if you know that you have some linear correlation methods will be good when you have linear relationship, when you don't have any nonlinearity, strange nonlinearity. So this, but I think this is, in my opinion, is maybe not so interesting because you already know, you are already guaranteed theoretically that you have a linear, you don't have nonlinear terms, correlations will give you the perfect relationships. So in that sense, I mean, we could have done the experiment, but I think it would not have added so much. Well, you're right, random forest will be good when you have some nonlinearities. So I think, again, what this is telling you, I think is not, this work is not so interesting, in my opinion, to tell you which method is good for each data set. For that, the Dream Challenge already, the original paper, did a very nice work. This is most to demonstrate that even when in more real data set, you're going to have a combination of linear and nonlinear effects. So maybe what makes more sense is to combine both a linear regression method and a nonlinear method. And then you are basically done. You don't need to know what is the best method for your data. So I think this is the conclusion to take here, rather than a study of which method, which this is done in the original Dream Challenge paper. So maybe probably you can go there and have a look at the results. Yeah?
And do you know what happens statistically in the return of the code? Why does it work so well?
Yes, actually, so they did some, try to justify why it works. They say that it's an example of the central limit theorem applied to network reconstructions. The first time I thought about it, I thought it was a bit strange, but actually, in the sense, if you think about probability or statistics, actually, with the central limit theory, what it's telling you is that when you repeat many times the experiment, eventually the mean of this experiment is going to converge to a Gaussian distribution. So actually, you don't need to know the particular distribution of each individual experiment that you know you are guaranteed to converge in the regime where you have enough experiments. And here, I mean, I know the analogy is a little bit wishy-washy, so don't take it as a theorem. But here's a little bit the same idea. You have an ensemble of weak learners. Each one have weaknesses and strengths. If you combine them, you are probably approximating a distribution that probably is closer to the real distribution. I think if you go to I think in the original paper in the Dream Challenge 5, they give a more rigorous demonstration than I'm telling you right now. It's not super rigorous, it's not a theorem. I'm not sure how you could demonstrate this theoretically, maybe it can be done. But they give you a little bit an intuition along these lines. It's like you are approaching the real distribution. And actually, this is what is done in machine learning all the time. All these ensemble methods, they're the same. They have an ensemble of weak learners. And I don't know if this is theoretically proven, but it's actually a matter of fact that it works every single time. You average an ensemble of weak learners, and you get a more robust learner. So I think it's what happens here. Each learner is capturing disconnected pieces of information, and when you put them together, somehow it works for your benefit. You reinforce those connections that appear in most networks, and those that appear only in a few disconnected networks, they are discarded. So you are converging toward the ground truth. Any other question? Okay. Yeah.
Still similar question. How weak are your learners? I mean, if they are really weak, there are many false positives. So the combination also accumulates the false positives. And is this possible that, and here we use the word wisdom of crowd. Is there such thing like stupidity of crowd? I mean, maybe it accumulates the mistakes.
Well, of course, they cannot be super weak. They cannot just have a random predictor and average then and expect that you get something real. So you have to have at least some learning. But weak learning, for instance, I mean to have a linear regression method, in the sense that with a linear regression method, you are going to predict a few things that are real, but because you are limiting yourself to linear relationships, you are going to miss a lot of nonlinear interactions. So that for me will be an example of a weak learner, that you are predicting some things, but you are missing many. So the nice thing of this approach is that you are complementing. So you have some methods that are linear, so they only identified a few linear relationships. You have Bayesian methods, you have mutual information methods that also have their range of application. You have this random forest decision trees method. So each one of them is good at pick up different predictions. So at the end, so each one is a weak learner, but of course it's not a random learner. You are learning something, and then when you are learning something, the aggregation of these learners will lead you to a better prediction.
Yesterday, I think Julio talked about another dream challenge that they have rarely results better than random. For this kind of weak learner, or maybe this kind of difficult question, do you think it's still we can still apply this combination?
So sorry, Julio presented.
I think another dream challenge.
Yeah.
There, most of the results are nearly the same level of the random.
And when they aggregated those, they got some real prediction? So you just said, so they have an ensemble of weak learners that are close to random? And when
not weak learner, I think it was because that question is really difficult that we don't have a good prediction results. So for this kind of question, would you think it's a good idea to apply this combination strategy?
So I'm not sure what he presented yesterday.
I don't remember the name of the challenge. I just remember that sometimes we can't. I mean, all the solutions are not much better than random.
And when you aggregate them, did he say something about the aggregation or not? Did he say something about the aggregation or not? He did not discuss this.
I don't remember.
Maybe we can maybe discuss later on if you want. Okay. So just as I said before, for this we also have a web service that is again free. You can use it. You can upload your data. Because some of these methods are computationally quite intensive. At this point, we limit to our not super large number of genes. I think here we have the limit of 250 nodes. Maybe it has been removed, but maybe you cannot run 5,000 nodes because some of these methods are computationally very intensive. But here you can select which network, which method you want to use. You can select all of them, and you can also select the consensus, the aggregation approach, and then you will get your prediction, and then you can download it as a graphical interface, or you just as a table where you have your predictions. So by all means, feel free to use it. And well, I wanted to say one word about this last pillar. This is not what we have done, but I wanted to mention because Julio was here yesterday. So I want to give a lot of advertisement for his group. They have developed this approach, OmniPath, where you can also parse a lot of databases available. They have they run it as a Python command, so it's very easy, and then you can parse and download interactions from these databases, and this is rather simple, so you can also integrate prior knowledge into your approach. This OmniPath is by Julio and his team. Okay, so if there are no more questions, I'm gonna, in the last still have like maybe 20-25 minutes. So I would like to move now to a different problem, to a different work. And what we are going to start, we're going to tell you a little bit about what we are doing about drug modeling. So when we started working in this, maybe into this a couple of years ago, but like we wanted to start with a rather simple approach. So basically, these are the two questions we are trying to address at the time. The first thing was a rather simple problem. It's like how to, again, thinking about personalized medicine. Suppose you have a patient with a particular biomedical profile, biomolecular profile. Let's say you have the transcriptomic profile of this patient, and you would like to predict what is the best therapy for this patient. That will be the first question. And then this, I'm not sure I'll have time to talk about this, but we are also thinking about how to develop new compounds. So let's see if we get there. So just to tell you from a machine learning, deep learning point of view, these are rather two different problems. So the first one is a prediction problem in the sense that you have a collection of available drugs and you try to match a patient with optimal drug or combination of drugs. The second is much harder. It's a generation problem. You have a patient with a particular profile of a transcriptomic profile, mutational profile, and you're trying to design the therapy for this patient. We are working on this, and let's see if we get there. Okay, so let's start from the very first project. This is what we call NetBite. So when we started working on this, at this time we were starting, because we were beginning, we started with a rather simple question. So let's say that you have this molecular data set. So you have this, for example, a transcriptomic profile. Here we're working with cell lines, but cell lines is a model that tries to approximate patients. So thinking about cell lines. So for each cell line, you know the molecular, you know the transcriptomic basal profiles, and you also know the molecular alterations, copy number alterations, etc. And then there are certain large drug screenings, like here we use the GDSC, which is a nice collection. It's a public data set where you have this characterization of these, I think, 1,000 cell lines. And then for most of these cell lines, they have been tested with, I think, 200 compounds. So for each one of them, you're going to have the IC50 value. So IC50 value is the concentration of a drug that you have to give to produce effect on half of the cells. So because these are anti-cancer compounds, that means to kill half of the cells. So this is the value that usually one of the values that is used to measure efficacy. Okay, so as usual, when you start at the beginning, the questions that you ask are not novel, in the sense that there are many people that have been trying to predict IC50 values using different machine learning algorithms. And one approach that is used quite often is random forest, because again, this is a nonlinear problem, so there are also people doing linear regressions, but random forest is like a nice approach in the sense that you have usually more variables than samples. So this is a recap, because I'm not sure you are familiar, or everybody is familiar with it, so this is a recapitulation of how random forest works. So in random forest, you have a selection. Basically, you try to select what are the genes that better predict their outcome. The outcome is the IC50 value, your particular experiment. So what you're going to do is like you have a selection of different trees, and so at each iteration, you select a subset of your genes. This is one of the controlled by this variable, and try. And then from this subset of genes, you select which ones are the better predictors. And then, according, you take this better predictor, and then you have a split in your tree, and then you continue. After you reach a minimum number of nodes on your leaf, which have been predefined by this variable, and then you stop. And you repeat this many times. One second. You repeat this many times, and then you have a decision tree at each time. Then you have, when you put all your trees together, again, each tree is a weak predictor, but put them together is a more robust predictor. It's called a random forest.
Yeah, so the IC50 values are the continuous values?
Yeah.
So you have to discretize them first, right?
No, you don't have to. So let me think about it. No, in general, you have to here. No, in general, you don't have to discretize here. What you're going to do is just select the genes, and then you try to predict. You can build a. Now I'm thinking about how we do it. Would you predict a regression on these genes? No, I don't think we have to discretize the IC50 values. No, I think. No, I'm pretty sure we don't discretize values. No. No.
Random forests are happy with.
Yeah, exactly. No.
More or less.
I was confused for a second. No. Okay, so as I said, this is not novel, so what did we do on top of it? So when we started thinking about that, when we thought is that random forest is great. It predicts quite well when you have a much larger number of variables than samples, but it has several limitations. The first of one is that it does not take into account prior knowledge. And we know a lot about drugs. So for some drugs, we know the known targets, and we also know the map of connectivity of molecular interactions within the cell. So what did we do? So actually, we defined basically what our idea was. Okay, I think I lost the battery, doesn't matter. So the idea is here we want it here in this random forest where you are selecting the genes using a uniform distribution. So each gene has the same probability of being picked in the next trial set, that is from which you choose the best selector. So what we wanted to do is just to bias this distribution such as genes that we know they are more informative because our prior knowledge have a higher chance of being selected. So this is, in a sense, will be a biased ensemble method. And actually, as I said, the prior knowledge here will come either from drug targets, so you know from each drug, at least you have an idea of some targets. Very often, your knowledge is incomplete in the sense that there are targets that are not known, or very often, sometimes this knowledge can be wrong, so you don't want to completely bias toward this prior knowledge, but you want to give this prior knowledge to the algorithm so it learns a bit better from it. And also, we do know the network of molecular interactions, as you can see here. So how did we do it? So this is what we call our biased ensemble method. So the idea is that starting from this is a network of molecular interaction that you can have inferred either using the method that I presented before, like interact or COSIFER, or just take your network, your preferred network, like string. So you have, for instance, just drugs, you have just studying drug X, and you know that these are the targets of this drug. So you can project these drugs into your the targets into the network, and then you follow what is called the smooth, it's like a network smoothness. So the idea is simple. You have your network, you know that when you perturb with this drug, this node and this node and this node are perturbed. So you are propagating the perturbation. So the first nodes would get a larger part of the perturbation. Then as you keep propagating through the network, the perturbation gets weakened and weakened. So basically, it's an iterative approach in which you just distribute weights to bias the distribution, where the drug target gets the larger weight, first order networks get a smaller weight, and then after a few iterations, basically the distribution is flat, and they get a smaller probability of being picked, but still they can be picked. And then you integrate this into random forest. So this is what we call net bias. So you integrate this network bias approach, and then the procedure to select the nodes for your random forest, and then you keep running random forest in the same way as before. Okay, so let me show you some results. Does it work or this does not work? Well, so our first experiment were a bit disappointing in the sense that it's what we're showing here. Here, and the y-axis, I'm showing the correlation with experimentally predicted IC50 values. So I would expect, in a perfect scenario where I get the perfect prediction, we get correlation 1. Of course, this is very optimistic. So at least you get to get a correlation as high as possible. So what we show here is that you can see the random forest in blue performs a bit better than net bias. So we were a bit disappointed at the beginning. But actually, this is only a very superficial characterization of the results. What we realize is when you start looking a bit more in detail about which predictions for which drug we predict well and for which one we do not predict well, actually we found something very interesting. For instance, here in this plot, I'm showing these are different family of drugs. So here you have, I don't know if you can read, but this is RTK signaling, which is a very known, well-known receptor. And then you have mTOR signaling, so these are different types of drugs. And you can see that here for RTK and EGFR, which are another well-known receptor, surface receptor, in these both cases, we perform substantially better than the other receptors. So actually that led us to maybe hypothesize that actually for these particular receptors, the prior knowledge is much more accurate in the sense that these are receptors that are in the surface of the cell. So you can expect that the targets are very characterized, and also this is a well-defined cascade of events. So actually the targets are more likely to be correct targets, while maybe you're looking at drugs that impact cells in the cytoplasm, it's a bit harder to characterize correctly the target. So actually what we look is we try to look at other drugs that also act on surface receptors, and we found that for all the three categories of drugs, that's what I'm showing here, RTK, EGFR, and IGFR, so it was the 3 families of drugs that act on surface receptors. In all 3 cases, actually, we NetBi performed better than random forest. So here, well, in this plot, you can show here the performance for different numbers of trees. So actually, you show that even for a very low number of trees, here you only have 10, which is actually at the very low minimum. You see that here, again, NetBi performs substantially better than random forest for these particular receptors. So actually, what that led us to hypothesize is just actually what, rather than just being a prediction, NetBi being a prediction to predict IC50 value for any random drug, actually it's a good tool to identify biomarkers. In the sense that when you have good and solid knowledge about what are the right targets of a particular drug, this approach can really speed up your computation. So this is rather than a tool for IC50 prediction, it's a tool for biomarker identification to know which drugs are good to predict sensitivity of a particular drug. Is this clear? Yeah? Or questions so far? Well, it's only in the case for the top plot. The others, I would say, not so much. In the top plot, yes, I give you that. You're right, the variability is larger, not in the other ones. Actually, I don't know. Sometimes the data behaves in particular ways. I mean, it would say that in the 3%, in this case, it's not the case. You have similar standard deviations here. Here, for instance, here you have a smaller standard deviation, and here is, let's say, comparable. So it's only in this particular simulation. So yes, it's interesting, but I don't have an answer to that.
And also, when you are using for the knowledge stream, again, you use the 0.7 of confidence in the threshold, or you try to go even to lower confidence just to try to annotate as maximum of interactions as possible. Do you have a difference in the performance of your method when you go to different confidence level of the stream?
So when we use a stream, we only use a maximum confidence level because actually a stream is a great tool, but it also can be a catalog of many low confidence interactions in the sense that sometimes you are combining high throughput experiments that might not be so reliable. In the sense like, I think for instance, there are different experiments that have different levels of confidence. Those that are really biochemical interactions that are done in a low throughput manner might probably be more reliable than a high throughput experiment where many things can happen. So when we use tools or databases such as string, we always take the maximum confidence. Otherwise, you start getting really networks that are not so reliable.
I think it also depends on your question, because if you really want to annotate and have information of as many nodes as possible, as many properties as possible, to later validate experimentally, maybe sometimes it's better to begin with as maximum information as possible and then later be more accurate, be more extreme, depending on the testing.
Well, this is a debate in the sense that what is better, to start with a high quantity of low confidence information or to start with a lower quantity of high confidence information? I tend to favor the second. Of course, it's a debate. I think just when you start with there's the saying that trash in, trash out. When you start with very low confidence information and you start processing and doing more computations and more transformations, you really have very little confidence on what you get at the end. So already this data is quite noisy. Stream is noisy, even the high confidence interactions. All these drug screenings are noisy. So we're not talking about super high quality data. Let's be honest about it. So it's better just to maybe have less predictions but with higher confidence than try to predict too much and then you don't have control on what you're predicting. 10 minutes. Okay, so now I have a doubt. I don't know if I should just go into tell you what we did after this with using deep learning, or I should just go into a different. Okay, since I only have 10 minutes, I'm just going to continue on that because I don't want to switch to a completely different topic. So as I said, NetBi was, this is actually something we did with Julio, so we know him well. So as I did, NetBi was the first approach that we did using this biased random forest, and actually was the NetBi we were integrating prior knowledge with transcriptomic information. So after doing that, we thought, okay, can we integrate more information? So for instance, at this point, we are completely disregarding the structure of the compound, but this is also quite a lot, very valuable information. So if you have a compound, and you can give to your algorithm or to your model the chemical information of this compound, actually you are giving a very valuable information to predict IC50 values. Okay. So actually what we started thinking about this, it was thinking right now so in the previous approach we use prior knowledge, in the sense that we are giving information about the drug targets, about the network of molecular interactions, and then transcriptomic profiles. So here we added this additional layer, so information about the chemical compound. And because now the system is a bit more complex, we decided to just leave random forests on the side and we moved to deep learning. So here I don't want to be this is a bit more complicated and it goes more into the realm of deep learning, so I don't want to be overly complicated. Let me just tell you a few things, maybe a bit with my hands, and then I'm happy to discuss in the coffee break if you have questions. So about encoding drug compounds, so this is a complex matter. There's not a unique way of, so compounds usually is a chemical, it will be the rather obvious way of encoding a compound, it will be through a graph. You could think the organic, the molecular is you could depict it as a graph. Because a graph usually is not easy to manipulate numerically, people have defined different encodings. So usually people use fingerprints. This is what this FB stands for. Fingerprints are usually, there are many different types of fingerprints. They are usually hand-engineered features. So for example, Morgan fingerprints is just people have defined that these particular chemical features are informative, and then you create your vector of fingerprints and you train your algorithm with that. So this is an approach that has been used quite a lot in the literature. It has several problems in the sense that these are hand-engineered features, and it's biased. It's biased by what people think is informative. It's biased in the sense that sometimes it might not be available for your compound. If you are designing a new compound, you don't know what are the fingerprints. And sometimes they are not easy to compute. But I think the most, for me, the biggest concern is essentially hand-engineered. So you have to define what is informative, and we humans tend to be biased. So especially with deep learning has the great power of being able to process raw data and extract information. So when you use deep learning, it really makes sense to give data as raw as possible. So this is what we're trying to do here. So instead of using fingerprints, well, we use fingerprints as a baseline for comparison. But actually, we focus a lot on using the smiles. So smiles are alternative encoding in which still is not a graph. That will be the next step. But it just resembles more a graph. So again, this is a very super, I don't have an introduction about it, but it's basically what you do here is C stands for carbon, another carbon, and then you have, this will be a covalent bond. So actually it's a much more, it resembles more the chemical, this is a way of encoding it as a text string, the chemical structure definition of the drug. So it resembles a bit more what is the chemical compound. And there are many papers that are showing actually that smiles are better characterizations than traditional fingerprints. So without going into the details, I just wanted to, so forget about the text, just telling you that we designed this deep learning model we call Pac-Man, and actually it has two different pillars of information. So this one is the one that is modeling, is just digesting this gene expression data. And we use a similar gene prioritization scheme as NetBias, so we also bias in towards the most informative genes. And this is the pillar that is really is digesting the chemical information. So we use mostly smiles representations, but we also use fingerprints as a baseline. So I'm not going to go into the details, as you won't ask me in the coffee break. And the nice things, again, as I will show, obviously now, because we have deep learning a kind of very reliable models, they operate with high accuracy, given that you have sufficient data. So actually, we kind of perform with high accuracy, and when we develop what we call the multi-scale convolutional attentive autoencoder. It doesn't matter. So we develop a model that actually outperforms all the others. But what is important of here is not really the particular model we develop what performs best. I think what is really important, which I would like you to remember of this part of the talk, is that we included modules that were attention modules, that are modules that tell you which patterns of your data were more informative to make a prediction. So basically, there is an attention module for the gene expression data pillar, and also there's an attention module for this chemical information. So that, for instance, when you get your predictions, so for example, these are two drugs. Here you have masitinib, and here you have imatinib. They are quite similar, so the only difference here you have a sulfur, and here you have a nitrogen. So basically, at the end, you get a predicted IC50 score, but then, interestingly, you get these heat maps that tell you how informative each molecular pattern of your drug was to make the prediction. And here, this is really cool, I find, because, for instance, you can see that in most of this part of the drugs are identical, and then here you get similar attention scores distribution, because these parts are unique. But where you find a big difference is here. Here, the sulfur actually here has a much higher attention score than the nitrogen. And that tells you that actually Bachmann understood that this part is unique and is what giving you the higher sensitivity to this drug. So actually this attention mechanism was very nice in the sense that yes, improves the performance, but also tells you afterwards which parts of the drug were more informative. And that allows you later on to try to design your own drugs, but maybe modifying the parts that are not very effective. You can do the same at the gene expression level. You can also look at which genes were more informative to make the prediction. Here we're looking at leukemia data, the leukemia cell line, and actually all the genes that we found that were given high attention are all related to either leukemia patient stratification or biomarker for leukemia, etc. So it was again a nice validation. And then once again we have the web service that is also publicly available, where this is an old screenshot. Now it has changed a little bit. So now you can also, you can either introduce your, when you log into Pacman, and actually there's no login, it's login free. When you get into Pacman, you can either select FDA approved drug, or now you have a drug editor where you can even design your own drug, and then you can give it to Pacman. You can also upload your own cell line profile or select one of the profiles from our data set, and then you get the prediction. So I'm running out of time. I just wanted to tell you, I don't even have the slides here, but I want to tell you what we are doing next, because this is already published. So what we're trying to do now is really playing with new developments in the field of deep learning, that's called reinforcement learning. So we're trying to now design new molecules based on a molecular profile. So that means what you have, suppose in the future you have a patient in the clinic, and you know this patient have these mutations, X, Y, and J, and Z. So you might start with a particular drug, but eventually you might want to optimize the patient, you might want to optimize the therapy for this drug. For that, there are certain approaches. One reinforcement learning in the ones is like kind of iterative approach in which you start with one drug, you give it to your, it's called a critic, it's the one that will tell you this drug works yes or no. If it does not work, you try a different iteration. It's somehow as a random walk in the space of drugs. And then you get as a reward, positive or negative, and then you keep iterating. And eventually you converge to something, and what we are demonstrating, at least at this point computationally, is that you are converging to something that is much more effective than the previous works. So I'm not talking about that because this is work in progress and also running out of time. But again, I'm happy to discuss in the coffee break if you have questions. And I think with that, I'm going to conclude. So I want to skip a bunch of things because I know we're super optimistic about what I can present. Now, I always put things because I never know what I'm going to talk about. So I did not think I will be able to present everything, but I put it no matter what. So let me conclude. So I present first, told you a little bit about what we're doing with single-cell data. I just presented one particular project, but we are actually actively working on the analysis of single-cell data, so I'm happy to discuss if this is a common interest for you. So actually, this is not in the right order, sorry. So I presented only the last project. Sorry, I thought I changed the slide, but actually I did not. So basically, I told you about dynamic graphical lasso. That is the approach that we have to explore time series data for robust network inference, and that in the sense that you can reconstruct your network and get a more robust inference using time series data. And then here I've moved after I left single cell on the side, and I moved to more traditional network inference prediction. Sorry, the animations are a bit messed up. I'm sorry about it. So basically, first I told you about Interact, which is the approach we have developed to extract information from word embeddings to predict protein-protein interactions from word embeddings. And I showed you how this performs better than traditional distances in these high-dimensional spaces. And then I told, well, I think this is, sorry, forget about this conclusion. I think it's completely messed up. I thought I uploaded the slides. So then I present a little bit COSIFER, in which I was telling you how this consensus interaction of these consensus networks could give you a more robust prediction for network inference. And the last part I think is correct. I told you about NetByte, which is this our bias random forest approach, in which basically we are biasing the picking up of the genes in the random forest algorithm, such that we are prioritizing informative genes and how this allows you to infer at least what are the biomarkers for drug sensitivity. And then I mentioned a little bit Pacman, a very summarized explanation of what we're doing on this drug discovery approach. And I'm happy to discuss with you if you are interested about any of these topics. And just I would like to thank the people who did the work, like the people at IBM and the collaborators, and also funding, because we could not do anything without funding. So thank you very much, and if you have additional questions, I'm happy to take them.